Hello everyone. This section is for cultivation methods of cellular microbes. After learning this section, you should be able to differentiate the streak plate, spread plate, and pole plate methods for isolating pure cultures. The streak plate method and similar methods for isolating pure cultures were developed by Robert Koch. The use of these methods transformed microbiology, and within 20 years of their development, most pathogens responsible for major bacterial diseases had been isolated and identified. Koch reasoned that if cells from a mixture of microbes could be spatially isolated from each other, each cell would give rise to a completely separate colony. Colony is a microscopically visible cluster of microorganisms in or on a solid media. Because each colony arises from a single cell, each colony represents a pure culture. Firstly, let's see streak plate. In this technique, cells are transferred to the edge of an agar plate with an inoculating loop. This is inoculating loop. The picture below shows streak plate techniques. In this technique, firstly, streak the cells over the surface. After the first sector is streaked, the inoculating loop is sterilized and an inoculum for the second sector is obtained from the first sector. A similar process is followed for streaking the third sector, except that the inoculum is from the second sector. Thus, this is essentially a dilution process. Eventually, very few cells will be on the loop, and single cells will drop from it as it is rubbed along the agar surface. These develop into separate colonies. On the picture on the right, you can see separate colonies. Then let's talk about spread plate and pole plate. Spread plate and pole plate techniques are similar in that they both dilute a sample of cells before separating them spatially. They differ in that the spread plate spreads the cells on the surface of the agar, whereas the pole plate embeds the cells within the agar. Both methods can be used to determine the number of microorganisms in a sample. For the spread plate, a small volume of a diluted mixture containing around 25 to 215 cells is transferred to the center of an agar plate and spread evenly over the surface with a stereo bent rod. You can see the stereo bent rod here. The dispersed cells develop into isolated colonies. This picture shows isolated colonies. The pole plate is extensively used with bacteria, archaea, and fungi. It is particularly useful when sampling a heterogeneous population of microbes, some of which might produce colonies that spread over an agar surface if isolated by the streak plate or spread plate methods. In the pole plate method, the original sample is diluted several times to reduce the microbial population sufficiently to obtain separate colonies when plating. You can see it on the picture on the right. The original samples is diluted several times. Then, small volumes of several diluted samples are mixed with liquid agar that has been cooled to about 45 degrees. And the mixture are poured immediately into sterile culture dishes. Most microbes survive a brief exposure to the warm agar. Each cell becomes fixed in place to form an individual colony after the agar hardened. You can see the isolated cells grow into colonies on the surface. They appear round, and within the media appear lens-shaped. The isolated colonies can be counted or used to establish pure cultures. If replicated dilutions are prepared and the number of tubes yielding growth determined, then the number of viable cells in the sample can be estimated by the most probable number method, MPN for short. Similarly, the number of viruses in a solution or the number of antibodies in a blood sample can be determined by first diluting the sample and then testing for the presence of viruses or antibodies. At last, let's see the microbial growth on solid media. Colony development on agar surfaces aids microbiologists in identifying microorganisms because individual species often form colonies of characteristic size and appearance. You can see on the picture in the middle. The colonies have different morphologies. From their form, the margin, and abbreviation. There are different forms. You can see the punctate form, circular form, and filamentous form. The margin are also different, and the deviation. There are many deviation types, such as flat, raised, and so on. When a mixed population has been plated properly, it sometimes is possible to identify the desired colony based on its overall appearance and use it to obtain a pure culture. That's all for this section. Here is a word list for you.